Plants face a severe mechanical disadvantage. They are sessile organisms, meaning they are physically rooted to a single location for the duration of their life. Unlike the animal kingdom, where organisms actively travel to locate mates and disperse their offspring, a plant remains stationary. This creates a difficult logistical problem. A complex organism must somehow transport its genetic material across hostile distances without moving its own body. To bridge this distance, plants deploy a specialized structure, the angiosperm flower. This is a transient reproductive apparatus. It is not a permanent part of the plant's anatomy, but a temporary tool used during a specific phase of the life cycle. This apparatus operates through a three-part sequence: manufacturing gametes, executing the physical transfer of genetics, and synthesizing a self-contained payload to be shipped away. Every petal, scent, and internal structure is a component built specifically to solve the physics of reproducing while stuck in one place. The structure begins with the thalamus. This is a flattened, swollen portion of the stalk that serves as the foundation for the entire reproductive machine. On the outermost ring is the calyx. This consists of green, leaf-like sepals that provide physical protection for the internal components while the flower is still in the bud stage. The second ring is the corolla, made of brightly colored petals. We group these outer two layers as support whorls because their function is protection and vector attraction rather than direct reproduction. Moving further inward, we find the third ring, the Androecium. This is the male reproductive engine of the flower. The Androecium consists of stamens, which use a long filament to support an anther. This anther is where the plant manufactures its mobile genetic payload, the pollen grains.
Finally, at the very center sits the fourth ring, the Gynoecium, the female reproductive engine. The Gynoecium acts as a central processing unit. It features a sticky receptor called the stigma, a conduit tube called the style, and a basal housing unit known as the ovary, which contains the female ovules. This concentric four-whorl architecture is optimized to manufacture, stage, and receive gametes, preparing the plant for the physical transfer of its genetics. Pollination is the physical movement of pollen grains from the male anther to the female stigma. Since the plant cannot move the pollen itself, it must hijack external kinetic energy. These external forces are transport vectors, categorized as either biotic agents like insects or abiotic agents like the wind. Flowers optimized for insect pollination must expend metabolic energy on attraction. This is reflected in the data. They produce large, bright petals, nectar, and scents. Their pollen grains are heavy and sticky, ensuring they physically adhere to the body of any insect that lands on the flower. Wind-pollinated flowers follow a different set of engineering requirements. These flowers abandon ornamental petals and nectar entirely. Because the wind does not require biological attraction, those structures are an unnecessary energy expense. Instead, they produce massive quantities of light, dry pollen and deploy large, feathery stigmas that act as aerodynamic nets to catch grains drifting through the air. A flower's appearance is a rigid consequence of physics, dictated by the aerodynamic or biological properties of its specific transport vector. The moment a compatible pollen grain lands on the stigma, it triggers a chemical process. The grain germinates and begins growing a pollen tube, which moves downward into the flower's tissue. This tube carves a conduit through the style to transport two male gametes directly to the basal ovary. Fertilization occurs when the male gamete fuses with the female egg cell secured inside the ovule. Once this fuse is complete, the flower initiates a teardown. The petals, sepals, and stamens, having served their purpose, wither and fall off. The central structure now undergoes a transformation. The ovary wall swells and thickens, developing into a fruit. This ovary wall becomes the pericarp, a vessel that protects the interior and eventually helps the plant disperse its genetics. At the same time, the fertilized ovules inside this wall harden to become the actual seeds. The floral display exists only to orchestrate this singular event, self-destructing once it has synthesized a durable vessel for the next generation. The resulting seed is a self-contained biological capsule designed to withstand hostile environments. Internally, the seed contains a dormant embryo, a food store, and an armored seed coat. Dicot store their food in two fleshy cotyledons, while monocots rely on an internal block of tissue called the endosperm. This payload remains dormant until it clears specific environmental gates: water uptake, oxygen, and optimal temperature. When these gates are cleared, the embryo activates and breaks through the seed coat, a process known as germination. Two primary pathways are used to breach the soil surface. In epigeal germination, the lower stem elongates rapidly, pushing the cotyledons completely above the soil. In hypogeal germination, the upper stem elongates, allowing the cotyledons to remain safely underground as the shoot grows upward. This completes the cycle. The new plant is now rooted in a new location, facing the same geographical isolation as its parent. A flower's architecture and its internal chemistry exist for one reason: to successfully provision and deploy this singular botanical payload across space and time.